So, welcome everybody to the session Preventing Cross-Contamination in Shared Facilities Equipment. So, a few information in the beginning. So, introduction of the session leaders. First is myself. I'm Richard Denk from SCAN AG in Switzerland, and I'm chair the session normally with Christa Myers, but she cannot make it today, so we have instead Lisa Dorn joining. A few words to Lisa. Lisa is with CRB, and Lisa has been in the pharma industry for over 30 years, guiding clients on innovative aseptic filling facilities, design and operations. So we have also other speakers this morning. This is David Ehertz from Allergan. That's Wolfgang Lau from Roche in Germany, and also Hartmut Schatz from NNE, also Germany. So there will be a 15 minutes question and answer discussion in the end of the session. So after every presentation, we continue with the next presentation, and then we have in the end a 15 minutes Q&A discussion. So we also the live stream. So we have also attendees from other countries, what we have heard this morning. So we have attendees from beside the English-speaking area from Germany, willkommen, from Switzerland, grüezi wohl, and also from Spain, buenos dias. So all of them are very welcome to joining the session. So I'm not only in the program committee and chair the track. I'm also the first speaker on containment. What I like to talk about is cleaning, GMP and cleaning requirements for highly potent aseptic products. So last year we had the first half-day track on high potent substance. This was such a trial that we see if there is an interest on the industry about those kind of products which coming onto the market and what have to be done to produce them safely. It was quite successful, and then we decided that we have this year our first full-day track about this topic. As mentioned, my name is Richard Denk. I work for SCAN in the position head containment, which means everything has to be done on preventing contamination and also cross-contamination. We have heard this morning a lot about isolator technology. So there's a lot also in the draft Annex 1 about leads to isolators. There's also mentioned about how to deal with highly potent substances, not much, but a little bit. And what is my topic for the presentation is a little bit to explain preventing contamination, but then what leads also to cleaning and cross-contamination. Because when you look at the new Annex 1 draft, there is a lot of mention of validation of cleaning. So what was the current requirements? Yeah? When we talk about aseptic isolators and factors for aseptic processing, it's in general mentioned cleaning and disinfection. That's get higher, as I mentioned in the new Annex 1 draft is mentioned, then this procedure should be validated, yeah, and this could be quite a challenge when it becomes to a very complex process inside of the isolator. And then afterwards we have an H2O2 decontamination. So this is in general an isolator well established. More than 80% of all isolators are decontaminated with vaporized hydrogen peroxide. So, but when it becomes now in the field on potent substance, we have a few more challenges. One challenge is that we have to produce mostly those products in a shared equipment or a shared facility. That means we have to have precautions, what we have produced before to the next product. So beside of cleaning, before disinfection, to remove any bioburdens, we have also to think about what was produced there, how toxic the product was, and what is the risk to the next product. And especially the risk to the next product is in aseptic fill finish, extremely critical, because there we have often the container as the dose. Yeah? We fill aseptically into a vial or a syringe, a product, and every contamination which gets into the container, it's in the container. So we have then afterwards still the same as decontamination with hydrogen peroxide, as I mentioned before. And a third part is also then operator protection. So there are people working in that field, in the area, close to the isolator or close to the product, and they have also to be protected from any exposure of this product. And the challenge in aseptic processing is that we have those isolators mainly in positive pressure due to GMP regulations. And we talk about containment levels, which in the non-visible area, but I will go to this a little bit later. Biotech. Originally, I must say I come more from the API and oral solid dosage forms, and I started with high potent products already 25 years back. So I know this from that area, what is done, and I still remember in my work in the oral solid dosage forms that there was mentioned this will be the major part in the pharmaceutical industry forever. And at that time also the biotech sector was more such a niche area, so vaccines and a few small biotech products were out. But when you look at those two graphs over there, the first one here I took from a source from the PDA, where they said that in the meantime, the most products coming from the biotech sector. This is, in my point of view, also the most innovative area with new drugs coming to the market. And this was presented, the second one, during the ISP annual meeting in San Diego in 2017, where there was mentioned where are most of those products are coming from. And when you look here, still we have a growth on vaccines and others, but the major driver, that means almost 50%, yeah, are oncology products, new oncology products, and they are mainly considered as high potent. Yeah? And this is something what those industry will face. We will face this more and more. So what are those kind of products? Yeah? Many of those are still in pipelines. So it's not that there is already many out there. The same with the left one with ADCs. There we will hear a little bit more later on the presentation. This is products where there are not many on the market, yeah? But you have to design the facilities already when you have it in clinical trials to be ready to use it when they are approved. And there is others like ATMPs, regenerative medicine, like gene and cell therapies, where they have a lot of products coming out, or cancer vaccines. So what makes them so critical? So when we look on such a monoclonal antibody, which is attached here with a warhead or a toxin, and this part here makes that combination so powerful and also so toxic, yeah? Because this is a general chemical produced small molecule which is linked to a large monoclonal antibody, and those normally designed to specially adapt to the receptor on the cancer cell, gets inside, and then it should release the warhead to kill the cell. Yeah? And those toxins, when I present in such conferences where there are hundreds of people talking about antibody-drug conjugates, they always say we have the most toxic substance ever on the market, yeah? Which is true. Yeah? They were too toxic to get them in another form applied to the patient, but most of them do not know how to deal with them and what it really means for the operator. So, in general, containment, as mentioned, and more than 20, almost 25 years in that field, was in the beginning mainly driven on operator safety. We have only spoken about how to protect the operators. This has changed in 2014 as the EMA released the guidance on setting health-based exposure, because this guidance made containment GMP, because then they ask or they require for each product permitted daily exposures. And then the toxicologists have to develop a lot of data for this, because it was first released in Europe, and in 2018 was it adopted from the PICS as guidance document. And so in general, all PICS countries, which the US also belongs to, will be inspected according to this guidance. Yeah? And why was it then updated? So in general, in years back, there was not a clear definition. It was a high-potent product. There was only mentioned certain products can be used in shared facilities. So this is now clear. And also the cleaning requirements, yeah, 10 ppm or 1/1000th of a daily dose. Yeah. Is this really suitable for highly potent substance? So now in the guideline is it clear mentioned, when you go in dedicated, that means when the risk cannot be adequately controlled. For instance, you have here a production facility with non-barrier system, yeah, where you can expose your operators or the room to the product, and you have not appropriate decontamination system before the operators leave the areas, or you have no PDE data. You cannot demonstrate how toxic your product is, then you go also in dedicated. Beside of beta-lactams, we are anyway, also the guideline mentioned that you are in dedicated. Or when you cannot clean to residue limits, that you can demonstrate that you clean in an appropriate way before you get the next product on the line. So, as mentioned, I'm dealing since many years in the high-potent stuff, and just to mention a little bit what could happen also when the ISB is producing guidelines. Risk map baseline guide was started, I think, in 2003 with the COP containment. Was then around 2006 first time introduced to the EMA. In 2008, then the EMA announced that they will revise their chapters. And in general, for the 2014 publication of the EMA guideline, is similar, the same mentioned what was mentioned in the risk map baseline guide. The only difference is the EMA guideline, it's mandatory, and the ISB guidelines mainly recommendations. So, to explain a little bit what we'll talk about, I would just like to start and get a little bit quickly through about decontamination of an isolator to get them from there into the cleaning and cross-contamination part. So, in general, you provide an atmosphere inside an aseptic isolator that you decontaminate the surfaces with, in general, with hydrogen peroxide. You have to design the equipment inside that it is possible to decontaminate. Yeah, this was also a learning curve in the industry many years back, how to design surfaces and the equipment, and you validate it in general with suitable biological indicators. So an isolator, I go a little bit quick through, this goes normally HEPA filters in, HEPA filter out, and you have a thermal HEPA filter which will provide you the unidirectional airflow, which is mentioned guidelines with 0.45 meter per second, plus minus 20%. So, and due to the high air, close a bit, then you normally recirculate the air. And this has to be a little bit different when we talk about high potent, because there I teach normally you have to isolate it in the smallest possible way. Yeah? Every spreading outside of a critical area might be not easy to clean, and spread products in other areas, maybe where other people like maintenance people could be exposed to it, yeah? And they often are not trained on how to deal with that. So then you start normally the hydrogen peroxide decontamination. This is normally done that you have here in 35% a bottle of hydrogen peroxide, you vaporize them, you get then the gas through the HEPA filter into the critical zone, into the isolator for decontamination. Generally also called flash evaporation, because the boiling point of hydrogen peroxide is higher than of water, and you have to get those hydrogen peroxide pumped with such peristaltic pump into the upper part of the isolator. On the heating plate, you flash vaporize it and get then the hydrogen peroxide through the HEPA filter inside of the chamber. Could be also performed in a different ways. You can also get it directly inside of the isolator. So those decontamination cycles are well developed, yeah. It's normally done that you make a leak test of the isolator first, make a preconditioning for temperature and humidity, and then you start the conditioning. That means you get the hydrogen peroxide inside of the isolator chamber. Then you have the decontamination phase where you decontaminate all surface, and then you start with the aeration of the area to get the hydrogen peroxide out of the critical aseptic zone. So I was recently asked, and I say, when it's always the question that in such presentations, that how innovative is the pharmaceutical industry? And I always say, when I go into the biotech field, I think it's the most innovative one. Because, as I mentioned before, I was many years in oral solid dosage form. On innovations were always mentioned. I say you have had an agitator for the blending, and the most innovative thing was that you redesigned the agitator. Yeah, this was then something what was then presented, that this is the best what you can get, and this is very innovative to do. Yeah? But I think within the biotech industry, when I look back, how fast all the disposables were implemented, how many of new products you develop, yeah, and which they come to the market and also sounds very promising for the patients. But the same here, yeah, when we look about decontamination of surfaces, yeah, the companies ask more and more, and extreme fast cycle times means very fast decontamination. And there is also a lot of development going on, instead of flash vaporizing with temperature, to using micronebulization, yeah, that you have very small droplet sizes generated with a spraying system, and you directly inject this into the chamber. Yeah? And there is quite important that these droplet sizes are in a specific range, and the smaller are, they are better for the fast evaporation, that they goes into the molecule phase. So you spray them in, they get very quick in the molecule phase, they get into very quick to the surfaces, and the larger one is also important, because you need a little bit until they get to every area in an isolator, that they are also vaporized and a little bit later. So the distribution is there quite important. But with a new technology, you can reach, in the meantime, decontaminations within a few minutes. Yeah? Similar here, when you look at this, there is the same leak test, there is more critical dehumidification. Then you get, in a very quick way, the hydrogen peroxide in it, on the surfaces. The holding time, as I said, it's minutes, yeah, then the surfaces are decontaminated. What have also the advantage that your aeration time is much quicker, because the H2O2 cannot be absorbed, isn't absorbed so long on the surfaces than the other technologies. So, cleaning validation for non-product contact surfaces. This is also a lot of work what I'm doing since many years, because it was not clear defined how to deal with that. Yeah? When I remember, as companies ask us and say, yeah, provide us a washing system for the filling line. And then I said, yeah, to what limit we shall clean this? And then I said, what do you mean to what limit? Because we clean visible, yeah? But what does visible clean mean? Yeah? This means only that you have removed everything what is visible on the surface. There's also in the EU GMP guidelines, chapter 5.21 of it, part 1 mentioned, depending on the contamination risk. Yeah? verification of cleaning of non-product contact surfaces and monitoring of air within the manufacturing area in order to demonstrate effectiveness of control measures against airborne contamination or contamination by mechanical transfer. Airborne contamination means of the potent substance inside. And we have in aseptic filling a lot of non-product contact surfaces. More than 95% are non-product contact surfaces, and some of them also critical to open containers. At that time, I set up an expert group where we worked a couple of years on how to define the rationale on cleaning close to open containers, which were also from toxicologists very famous from the company Roche, and GMP experts, and also a GMP inspector we could get for our document. Because this GMP inspector mentioned there, Rico Schulze from the German inspectorate, he's very focused on cleaning and cross-contamination. As I told him what we are working on, he said, I would like to join your group because this is actually really a topic that was not covered so far. So then in 2017 we published this, and to give you here a little bit a short introduction, what this means. Everything what is gray here, this is something that you can visibly see. When we talk about a stainless steel surface, highly polished, and then we consider normally everything below 400 micrograms measured on 10 to 10 centimeter as visible clean. And this is the same what you can see in the area. And when we leave that gray part, go to the yellow, then we have already reached the non-visible parts, also non-visibles on surfaces. And many of those toxins, they are on this level. Not all of those toxins, there's even a higher one, And in the meantime, I must say, I have to increase the containment pyramid even to a 7 span, because there is so many new developed biotech products like using for Botox, nerve toxins, which even goes into a picogram level, And just to explain you, we are here 10 times better, 100 times, 1,000 times, 10,000 times, 100,000 times. We must be partly 100,000 times to 1,000,000 times better to protect the operator or to protect the next product from the previous produced product. So in this one, we were coming up with such a table in this document, and based to information on the publication was publicated from the PDA in 2017. It's the most downloaded document what they have ever published so far. And where they are interested, especially in this table, where they say what are the limit surfaces for non-product contact surfaces within isolators based on GMP and occupational safety criteria, which have given there a limit. And the limit to define this gets more and more critical the more we leave the area of the stainless steel polished part, especially when we talk about cleaning of polymers, where the surface is not easy to validate. So we have the same here of limits of public surfaces with uncontrolled, more driven by occupational hygiene criteria. The reason for that is because people working there, and when they work in that area, they have uncontrolled access to contaminated surfaces. And this is also the reason why these numbers are 10 times lower than this one here, because people move around and can get in contact with the substance and can uncontrolled leave the area to other areas. And then we have also mentioned limits of airborne API inside of isolators after cleaning to avoid also that there is due to airborne contamination or contamination risk to the next product. As I mentioned to you, this is a new area as where we talk not about cleaning and cleaning validation as the industry is known it from batch size to the next batch size. We are talking here in areas where you have the dose for the patient, where you do not have any dilution factors afterwards. So this was published with the PDA letter, and I also introduced this last year on the conference the first time. Was then mentioned from Pink Sheet afterwards, what we have done here on this document. In the meantime, is it translated in German and also in Japanese. So it was introduced in Japan also a month ago, because this was there so high interest how to deal with that. So what else is coming? There will be another document about cleaning within lyophilizers, because there is also a high potential risk. You have open vial during the whole process where you get the vacuum on it, where you release the vacuum, and this is also a source of contamination. So this will also coming up to the industry soon. And other documents will also follow on how to protect the product and also the patient. Yeah. So for those of you who are interested in more detail on containment, this is also one of my babies. I published in 2015 the ISB DACH containment manual with my local expert group. So we have this all ready in translation. We work on the next version also together with David, our next speaker. And what we're also proud of this document, this is the only document which can be found in the library of the EDQM, of the European Directorate of the Quality of Medicine and Healthcare, and the EDQM also published the EU Pharmacopée. So with that, I'm on the end of my presentation, right on time. As I mentioned to you, question answers will be in the end of the session. And I will, one second. So I introduce the next speaker. It's David Eherts. Dr. Eherts joined Allergan Pharmaceutical in March of 2014 as Vice President of Global Environment, Health and Safety. Prior to Allergan, David has worked over 30 years in the field of safety, environment, health and safety, starting as a toxicologist in preclinical drug safety assessment at Merck. And he moved to environment, health and safety and has held position increasing responsibilities at Merck and Sanofi-Aventis. This presentation is really of interest, because many companies always say, containment costs only money, but it could also save money as you reduce it to the smallest possible way and just avoid the spreading of material. Thank you, David.
Thank you. As a good German, of course, Richard is precisely on time when he finishes his presentation, so I appreciate that. We're going to switch gears a little bit now. Richard, I think, has impressed upon you, as he has me, that his DACH affiliate are the technical experts when it comes to containment technologies, how they perform, and not just how they performed, but keeping them all up to date. So I really recommend his publications for that purpose. What the Center of Practice brings to the discussion is how to drive this technology into practice. So we know what works, but how do you get your leadership to make the investments and bring your company along? I thought we had a great introduction to this topic from Rick Friedman, and I'm being very careful not to paraphrase or quote him, but the impression he left on me was that FDA looks at your company and judges your company and your leadership by the technology that you put into place. It means the company's making these investments, and it changes the way maybe that the regulator looks at the company. And if you want to talk about value, that's huge value. Now, a little about my background. So I started at Merck. I like to say that even though I'm a vice president, that doesn't mean I don't know what I'm talking about, that I actually do have a technical background. So I started in toxicology at Merck in preclinical, and I moved from there to occupational and environmental toxicology. So that group set the PDEs, set the OELs, set the bans that Richard was describing. In fact, I've set hundreds, if not thousands, of bans at Merck and at Sanofi. And I'm doing it right now, too, at Allergan. So I've got an expertise in that, but I also teach at night at Tulane University, a number of courses, but the one that gets the most attention is called Financial Aspects of EHS Management. And what that teaches these young professionals is how to get their leadership to make the appropriate investments and the technology that saves lives. So I'm going to discuss a little bit about the benefits of containment from the point of view of the patient. Obviously, patients want high-quality drugs. They want the potency to be what the label says it is. They don't want recalls. They don't want contamination. Obviously, there's benefits to the patient in controlling your process. I think Rick made a great point about the value to the regulators. It makes it easy for them to regulate our profession, our industry, if we use the correct technologies. I want to talk about workers, too. That photograph on the left comes from a facility that no longer exists. And so it probably doesn't exist because they were using that particular process. In fact, when I first saw that photograph of them scooping the formulation out of the bottom of a fluid bed dryer, I asked why they weren't using a spoon. The very small scoop took them a very long time to do that. But most importantly, as part of that process, they were losing the formulation to the air in the room. And if you put ventilation directly behind them to try to keep the powder out of the breathing zone of the worker, you actually pull the product away. And what's even worse is if the API has a different particle size, has different polydispersal aerosol characteristics than the excipients, you could be preferentially pulling away API and leaving excipients behind, or vice versa. So it not only affects yield, but it affects conformity. And so that is a horrible process. And when activists moved that process from Corona, California to Manatee, Puerto Rico, they decided to make the investment in the containment technology that EHS was proposing. And I'm going to show you the huge savings that came from that investment, not just to the patient, not just to the regulator or to the worker, but also to the environment. And it brings value to the company. I talked to Rick briefly about the fact that technology like this increases the speed at which you can do research and development. So it gets your products to market faster. And then I'm also going to show you, as part of the cost-benefit analysis, that it decreases turnover times between batches. It decreases the amount of time employees have to spend gowning in and gowning out. And I think there's value all up and down the chain to the company and the shareholders. So we kind of divide it up into small molecules, oral solid dosage. I know this is an aseptic conference, but there's things to learn from OSD. And then the liquid, the aseptic process also benefits from containment. And most important is the pilot plant. One of the first things we did at Allergan was bring the pilot plant up to the latest technology. And I did that from a selfish point of view because I knew if we developed the processes using contained technologies that they would enter into production that way and I wouldn't have to worry about retrofitting later. So investments in the pilot plant pay back even more than investments in operations. I stole this slide from our site in France, and they stole it from a guy named Todd Conklin who talks about a new view of safety. And what he basically says, and I think it applies to quality even more, but we have certain processes that we know are fragile. They're often out of spec, and they're non-robust. And so it's pictured here as a table with a ball on top, and that ball is going to roll off that table very easily. And as you make investments in containment, the processes get more and more resilient, more and more robust. So stable is where most of our processes are now, where there's some open steps and some closed steps. And as long as we follow procedures, as the FDA knows, everything is okay. But the smallest mistake can drive that process into problems. And then the best processes are the ones that are resilient. They tolerate small mistakes by operators. I often say that if you've got a contained process, it prevents the powder from getting into the breathing zone of the worker. It also keeps the worker from getting to the powder or the product. You can't drop a pen in a batch that's fully contained. You don't have to worry about diversion of opioids from contained processes because the employees can't get to them. You don't have to worry about taking the pockets out of their uniform so they can't steal tablets if they can't get to the tablets. And so what we're shooting for here is resilient, robust processes, and I think as a direction, nobody can argue with that. So in this baseline guide that we're developing in cooperation with the DOC affiliate, we want to document the value that comes from containment. And it starts because, you know, I'm a board-certified industrial hygienist, so my first concern is the patient, my second is for the employee. If we can eliminate PPE, then we eliminate the potential to make a mistake in wearing PPE. And we know that workers often, because PPE is uncomfortable, and I was guilty of this when I was running long-term clinical trials of very potent products back in Merck days. If the room is very warm and you're wearing a full-face negative pressure respirator for 8 hours, you literally end up with a puddle in the bottom of the respirator. So the easiest thing to do to make it more comfortable is just loosen that bottom strap. And then you can get some fresh air coming in without having to come through the filters, and it makes it much more enjoyable to do your work. But of course, you're exposing yourself to the product. Well, if we had a contained process, you wouldn't need the respirator in the first place, and you wouldn't be tempted then to take shortcuts around use of the respirator. And I'm also going to show, and I think you'll be surprised, I've run into GMs before that have said a respirator costs $35 and a containment system costs $1.2 million. And I said, you're underestimating the cost of a respirator. If you've got a respirator and you're compliant with OSHA European standards, you've got to have medical clearance, you've got to have annual training, you've got to be fit testing, you've got to be changing the filters on schedule. You should be taking the Tyvek off the cal of a PAPR every time you exit a room and go back in the next time. And these costs add up dramatically. And if you can get your employees out of a respiratory protection program, first you can put them back to productive use, making pharmaceuticals, high-quality pharmaceuticals. But secondly, then you don't introduce errors. And then, of course, increase in yield and product conformity. I'm going to show you, we did a retrospective analysis of that process in Corona, California. And I'm going to show you all the waste that occurred because we didn't have a contained process. And as I talked about before, if you pull away the API preferentially to the rest of the larger molecules, particles, you lose conformity too. Obviously, decreased potential for cross-contamination. If the product's not on the floor or in the air in the manufacturing room, you don't have to worry as much about directional airflow and differential pressures because the product's contained within the unit. And my boss hates when I say this, but my goal is to be able to run the process in the parking lot because the environment you were containing is inside the equipment. It has nothing to do with the room you're working with. We know that's not exactly true, but directionally it is. Something I worry about from the safety perspective is the potential for dust explosions, especially in OSD. If you've got contained transfer, vacuum transfer, and you transfer as a bolus, you've occluded oxygen, and because the transfer technology is internally bonded and grounded, you can't have an isolated conductor. You can't get the difference in potential of electricity between different pieces of equipment that generate the spark that ignite the explosion. So from a safety perspective, we love to see vacuum transfer over manual transfers, or even dumping from a drum with a cone clamped on top of it, which often has a problem that when you open the plastic bag in the drum and you pull the bag over the drum and then you clamp the cone in place, you've isolated the cone from the drum, so you can ground the drum to the drum dumper, but you've got an isolated conductor in the cone, and as the powder goes through, it generates a potential, and that leads to a lot of explosions in the industry. You've got decreased loss of potential of the API to the environment. I've gotten involved heavily in pharmaceuticals in the environment, but especially antibiotics and antimicrobial resistance issues. I know the FDA is just interested in that too. And it doesn't just come from the physician over-prescribing, it also comes from poor manufacturing processes in developing countries. And the API ends up on the floor of the manufacturing room, it's rinsed down into the drain, ends up in surface water. It's been documented near these plants, and these NGOs are finding them and then putting pressure back on the manufacturers. If the process is contained, the powder can't get on the floor in the first place. So we think it has a lot of benefits to the pharmaceuticals and the environment issue. There's decreased potential for quality issues due to particulates and loss of asepsis. So if you don't have people in the room, they're not generating particles, they're not generating virus and bacteria in the room. That helps quite a bit, but even if there was some, and this is dangerous to say, but still the product's isolated if you've got a containment system in place. You've got decreased need for excessive ventilation. You don't need 20 room air changes per hour if you're not trying to decrease the particulate load in the room. And so if you're not generating powder in the room, you might be able to make some energy savings. And then I think, and this is underestimated normally, there's huge increases in productivity due to shortened or eliminated changeover times. One of the things I proposed doing in the past, it's a very simple technique, is to put a Petri dish in the corner of the GMP area. And every week or two, take that Petri dish, shoot 10 meters of methanol in it, swirl it around and shoot it on HPLC and tell me if there's any powder on that Petri dish. And if the containment technology is working as expected, there should never be. That's one way just to keep an eye on things. You've got faster transfer between unit operations. That scooping process that you saw takes an awful long time. Vacuum transfer takes just minutes. And there's decreased time spent gowning and degowning. Every time somebody goes into or out of the GMP suite, they have to go through a very long, arduous process to come into that area in a GMP fashion. If, number one, containment decreases the number of times you have to go in the room, and if you don't need the same level of barrier clothing on the individual, then you don't have to spend as much time gowning in. So I've got a couple case studies here I'm going to run through very quickly. I want to demonstrate that we have a robust process to do this cost-benefit analysis. So some of the slides are going to be eye charts, but I put them in here to leave you with the perception that it's a very robust, very precise system we use for doing the calculations. So this is the software that we use, and this is software I use when I teach EHS people coming out of university how to demonstrate to their leadership that everything they do for the safety of the employee actually saves the company quite a bit of money. So this just basically says all the reasons why you should be doing containment. This tells you the different aspects we're looking at. For example, we're looking at the amount of time wasted for operational personnel time to get in and out of the gowning room. We're looking at the amount of time spent on training for respirators when you don't need respirators. We're talking about loss of raw materials, product yield because you've got open systems. And then just to say this is a 10-year analysis with a 7.5% discount rate, a 2% inflation rate, and a 37% tax rate, obviously in the US. And the fully loaded hourly rate of a typical operation employee, not at Allergan, but it's in the industry, is about $40 an hour. And then what we do here is we just put out in English what we're looking at as part of the analysis. And I'm going to show you how we put these details into the algorithm. So the cost for moving that process, and this was quite a while ago, and these sites don't exist anymore, so I can say it freely, was about $1.2 million investment in the kind of stuff that Richard sells. The benefits, though, are, first of all, we got improved product yield, and we didn't know this until the first year was concluded. And the general manager at that site came back to us and said that essentially the yield went from the low 90s to almost 100% because, of course, the process is fully contained. And when he added it up for the year, it came out to be about 5,000 kilograms of an API. And then I wanted to take credit for the value of those tablets on the market, but they gave me credit for the cost of goods. So about 3 cents a tablet is what it cost us to make that particular product. But we saved, by the site's calculation, 13 million tablets. So that goes into the calculation. And then, because this site was in Puerto Rico, and if you're using single-pass air and doing high numbers of room air changes, you're basically taking 90-degree air with 90% humidity, cooling it down to 60 degrees, taking the humidity out, and then blowing it right out the window. And you wouldn't run your house that way, yet we run manufacturing facilities that way all the time in the name of quality. So again, we're going from this to that. This is all we're doing. And then if you want to look at other benefits, so you don't think of PPE as being very expensive. I mean, a single Tyvek bunny suit doesn't cost very much. But when you've got 30 people using 5 or 6 of these a day, and if they're doing it as per most of our procedures say, they're throwing them away as they come out. You know, we're not trying to reuse those. And if you're doing the same thing for the outside of a PAPR, and you're buying booties and gloves every time you go in and out, that adds up. And I was very surprised to see when we did the math, it came out to be very, very high numbers of PPE, in the hundreds of thousands of dollars. And then you get benefits of eliminating this time, scrubbing into GMP, and coming out and throwing all the stuff in the appropriate containers. And though it only seems like 5 or 10 minutes at a time, when it happens 4 or 5 times a day, and more often if you drink 3 cups of coffee before you go in, these guys are constantly going in and out, they're using a lot of PPE. They're also spending a lot of time getting dressed and getting undressed when they should be making product. And if you add that up across a fairly large size site, it turns into a head count or 2 of wasted productivity. Right? And then we didn't even include savings and changeover times because we decided to do the exact same amount of cleaning between batches, which I felt was unnecessary, but quality thought was the safest thing to do. We still cleaned the room with the same tenacity as when there was powder all over it. I would argue maybe you don't have to do that. And then, of course, you get products to market faster, and we didn't include that in the calculation. But just to go quickly through, so it does cost $1.2 million, and that's depreciated over 10 years in a straight line method, so that's $108,000 a year. Not that you spent because you spent the cash up front, but that gets charged against the site as a cost of goods every year. And a lot of GMs are being measured on their cost of goods. So they don't like making these investments if it means that their metrics financially at the site are going to worsen. But I think I can show them benefits way beyond depreciation. So if you look at it this way, if you just do that 20 minutes a day times 30 employees times 244 days times $40 an hour, you're saving, in the range of $100,000 in operational personnel time. If you look at the EHS supplies per employee going in and out all those times, you're saving about $400,000 a year in PPE supplies by just going to a lab coat and a hairnet and safety glasses. If you look at that 5,000 kilograms just at 2 cents per tablet, you're saving about $500,000 in cost of goods for the API. And finally, if you can reduce just 4 room air changes per hour in Manatee, Puerto Rico, you're saving the company about $100,000 in energy costs and contributing to less greenhouse gas, which all the millennials like, which decreases your turnover rate. And then this software that we use actually does the calculations for you, and whenever I get a raised eyebrow from the CFO or from one of our accountants, I take them to this screen in the software and I let them pick any one of those points and do the calculation on their calculator, and it's precise. So the bottom line is that making that $1.2 million investment resulted in a net present value to the company of almost $4 million. And I had a COO at the time that had some challenges in making budget, and he asked if we could count on that money. I showed him the analysis and convinced him that we could. It's got a 55% internal rate of return, 100% ROI, and a discounted payback period of 2 years. So I've got 5 minutes left. I'm just going to show you the same thing from the aseptic side. So this would be a site that we have that's considering this investment, and I did the exact same analysis for them. This basically is weighing in an isolator, transferring it through containment into the mixing vessel, and then adding the diluent, get the mix started, then out through the wall into the RABS unit. And this is all done in a contained fashion now. And so it's exactly the same costs and the same savings for that process, though because the API is much less quantity, you don't see the huge savings based on increased yield. But you do see the savings multiplied when it comes to getting in and out of an aseptic area versus a regular GMP suite. So I'll just go through these quickly and show you that even for an aseptic process, it saves close to $1 million in net present value to invest in an isolator and split butterfly valve technology to get the API into the mixer and then out into the RABS unit. So I've got 3 minutes left. I'll just finish up with a summary here. I would argue that having the information that the doc group generates on the exact performance of the technology is very, very important in making the proper choices and convincing your management to make the investment, but equally as important is this value proposition. that if you can go back to your management and show them that on a day-by-day basis they're saving money, they make the investment and sooner, and as I say, there's no buyer's remorse. They know they're doing the right thing for the patient, but how do they justify it to the CFO? And I think I've given them the method to do that. I know we have time for Q&A at the end of this, so why don't we hold questions until then? And I'll give you 1 minute back. Thanks.
Thank you, David. I could even now say, are you Swiss? Because they normally schedule it, they are normally faster as scheduled. So, but it's a very good presentation, I must also say. When we talk about the next presentation, also about new of those biotech products, often the value of such a batch, when I ask them companies what does such a batch cost, this goes often in millions, up to 100 millions per batch. And when this is then lost, for some failures, what is then, I really say this is one batch loss cost maybe the same as the whole facility to build. So the next speakers, I think you both come on to the podium. It's Wolfgang Lau, you're standing already. Wolfgang is a master of mechanical engineering. He started to work in 1986 as a project manager, working on the first fermentation project of Boehringer Ingelheim in Germany. Boehringer Mannheim, okay, sorry. After that, he started working mainly on sterile fill finish project for Roche. Yeah, that's right, in Mannheim as a project manager. So in a second speaker, I have Hartmut Schatz. Hartmut is a master's degree of pharmaceutical engineering from the University of Applied Science in Sigmaringen, as a more southern part of Germany, but very well known. Since he started to work in 1991, he was responsible for the planning and commissioning of pharmaceutical plants in more than 15 countries. Presently, he is working with NNE as a process expert for small volume parenteral products. And the presentation will be that's what we have heard this morning about ADC, high potent aseptic filling. Thank you.
So, hello everybody. As I was introduced, we will speak today about the ADC high potent product filling in Mannheim, and we will do it together with Hartmut. So, first of all, a table of contact. We have a short introduction for the project scope and the timeline, the products, the terms and definitions. And the most important part is the containment improvements and innovations. We will talk about the SHE risk analysis we did, the engineering approach, and general improvements. And the most important part is the primary containment improvements. These are details, how we solved certain problems. After that, we will talk about conclusions and recommendations. We've made some good and bad experience. We have some numbers, our contact, and we have a short video at the end. So here you can see our production site in Mannheim, and where you see the arrow there, this is the plant we built. This plant has 5 floors. We have 2 basement floors with change rooms and technical rooms for clean media, and also cold storage and small production. The main floor is the production floor on the ground floor, and above that we have a very high technical floor for air conditioning, mainly for air conditioning. And here you can see a cut through this facility. We have the 2 basements. And here you can see the freeze dryers with the condensers, the freeze dryers, and here you can see the distribution of the air ducts, and up top here you can see the freezing units and the air conditioning units. So this is a layout of the ground floor, and you can see different areas, the compounding and the area for cleaning equipment. Then what we mainly talk today is about the vial filling and the freeze dryers. and we have a third area where we do the inspection. So first, I want to introduce to you the project timeline. We started the CD phase in September 2013 till 2014. Then we had a stop of the project, and because the approval was in December 2014, and we ordered, then everything had to go very fast. We ordered the equipment in February 2015, had a short BD phase till June 15. Then we started construction of the building in September, and all equipment, major equipment was delivered in March 2017. So then the commissioning ended end of 17, then we did the IQ/OQ, and handover was in October last year. That means OQ was completed. At the moment we're doing PQ. Some of the PQs have already been completed, and we plan to start commercial production in 2020. So Hartmut will continue.
We will make this a little bit interactive, so we will change quite a couple of times talking about the subject. I mean, this was already very nice introduced. I mean, this is the product we are talking about. It's an ADC product, so basically it's a very high toxic product. I will come later to that point, like Richard has explained it. We are here working in a very, very low, this is actually the red marked area, in a OEB level of 1 microgram or less, what these products are about. And in order to illustrate this, because usually what is 1 microgram? Yeah, this graph shows this, I think, quite nicely, because otherwise this figure is quite difficult to understand. If you would take this very famous building, it has approximately a volume of 1 million cubic meters. And just from the break, I got this sweetener, and this sweetener contains 1 gram. If you would dilute this 1 gram evenly in the air of the Empire State Building, you would get 1 microgram. This should just show you, I mean, how much you have to do in order to secure this kind of containment, let's say safely, to have a safe place for the operators working in this facility. Also, this was mentioned quite a couple of times. We made a lot of thinkings in the beginning how we can elevate this facility up to a next level of security in order to enhance that what was currently already established in the industry in this new facility. And in this, let's say, first thinking what we did, we came to the conclusion that for sure we don't want to have personnel operating with personal protection equipment. We want to have a primary containment, which is a local containment, basically an isolator or a closed system, which is maintaining during normal operation a safe place to work. So therefore the primary containment design goal was to establish such a system that enables that all the operators can work in this facility without wearing any personal protection equipment. But of course, you also have to consider that something can go wrong, and therefore we have established what we are calling a secondary containment, or a second layer is, in case something goes wrong, either by operator mistake or by technical errors. We wanted to have, let's say, around the primary containment, a secondary containment, which ensures that, for example, if we have a larger breakage and an exposure of a high potent substance in the room, that this contamination is not spread out in the entire facility and therefore remains in there. Of course, in that case, people have to go in with personal protection equipment to clean it up, but we are sure that it will be not spread out furthermore. We did this in a quite extensive SHE risk analysis. We did this first in the conceptual design, but then very, very intensively and thoroughly also with all the equipment suppliers in order to discuss all the technical details, how this containment can be solved best. Also, we did quite unusual for this compared to other projects. We made a very intensive pre-engineering study. Basically, we selected 4 vendors, 2 for the freeze dryers and 2 for the filling lines, and made a very extensive pre-engineering, because we said we cannot, let's say, write down just a user requirement specification, hand it over, and then we get quotation, then we order it. Since we want to establish a new level, we really had to work with them together in order to achieve also a consolidated work with the supplier together, what such a system should look like and what should be the performance then at the end. Then the second phase was then the tendering and the contract awarding. So we made a very fair game. We give the supplier money for that, because you cannot expect that suppliers work with you for such a long time without, let's say, getting any money for it. So it was a fair deal right from the beginning. We said, okay, you get a pre-engineering fee, and certainly those who were selected and got the order, this pre-engineering fee was deducted from the contract value, but also the other one who has not got the order, they get at least the pre-engineering money, because they, of course, have to appoint, let's say, their most qualified people in order to do this very affordable work with us together. After contract awarding, certainly we did not stop the work. It was even more intensified. We had weekly meetings with the filling line supplier and also the freeze dryer suppliers in order to ensure that during the design phase we once again revisited the EHS issues, certainly also the GMP issues, to secure that everything would then at the end work to a safe system. So this is it at the end, and this is a very, let's say, a rough picture of the filling line, where you see, we call it lobster at the end because of the shape, yeah? That was consisting out of 3 freeze dryers, and different colors. What you see here are different isolator sections. So we said right from the beginning we did not want to have, let's say, a single isolator system, because this is not controllable. So we had a segregation right from the beginning in order to minimize contamination. Anyhow, it was the prime, let's say, goal right from the beginning to avoid contamination, and if we have contamination, to, let's say, minimize the area which will get contaminated. We will show later some pictures of that, how we achieve this, in order to minimize cleaning, which is costly, time-consuming, and not easy. In regard to containment improvements, I cannot go through all these issues. If you look at we call it action list, which is basically a minutes of meeting protocol with the suppliers. They are up to more than 1,000 points, for example, for the filling line. We discussed quite a lot of issues. For example, I just want to take out one. We, for example, have completely avoided any kind of vacuum systems on our filling line in order to avoid that the vacuum system would get contaminated, which we have to clean then afterwards. And also these 8 other points which are mentioned here were machine optimization, which was not there before the project, and which we established then together with the equipment supplier to enhance it to a next level security. We call it high potent 2.0. So then it's up to Wolfgang to show you, let's say, some containment improvements. I have to say we are very limited in time, and we will show you, I think, now 9 things a little bit more in detail, where we definitely think we have gotten improvements to the design which was available before. If anybody of you has a further more interest to deep dive in one of the topics, you are more than welcome to contact us. We are here today and tomorrow, and then we can discuss this offline. But time is limited, unfortunately. Yeah?
So one of the improvement possibilities is to use disposables, because you don't have to clean them. Very simple, and that's what we consequently did. We can use this line also with stainless steel vessels, but so we can use it with disposals or with stainless steel. So, but we decided to have every fluid path, and you can see the different colors as a disposable, and we don't have to wait along and do sip cleaning. We simply throw it away. So this is the main thing here. Here you can see one of these disposable. This is the most important one. This is the filling disposable, and you can see this is the wall of the isolator. This is what's outside of the isolator. The disposable is brought into the isolator with this bag with the alpha beta port, and this is the filters and the needles, everything what we designed for the use in the isolator. I think it's a very important detail if you use isolators for high potent purposes. So another detail is how do you clean such an isolator? So we're mainly cleaning with purified water. We could also use some zip fluid, and we wash all the surfaces of the isolator and also the ducts. So the outside ducts here, they have nozzles, and they are also cleaned after every process. Then after that, we dry the isolator with pressured air. Then we have no risk anymore for the operator. Then we can open the isolator and do a manual cleaning and format change. Then we dry it with the HVAC, and then we do the hydrogen peroxide decontamination. So here you can see another detail where we have the bag-in, bag-out filters. They're directly connected to this outside duct, and up to this flange we clean the whole isolator. So this is a picture of our freeze-dryer loading system isolator, and here you can see, for example, we pull out the air, this outside ducts, and this is also the pusher for the freeze dryer. So now I want to tell you how whole airflow of the system is. Here you can see the fresh air handling systems. There are 3 HVAC units above each other. Then you can see the same picture again, where we go in with the laminar flow above, outside channels, and then the ducts out to the isolator. Then you can see the bag-in, bag-out filters. This is a very important detail in our feeling, because these open to the clean room. Because if one of these double bags tears in the technical area, we think it's a very big risk for the facility. And so we decided to open these bag-in, bag-out filters to the clean room, because if something gets spilled there, I think it's a very much lower risk. Then we have an exhaust air fan, and we have an air handling unit for the exhaust air. This is only necessary to recover the energy. So this is another detail to ensure the pressure in these 2 important areas, in the tunnel and in the outside washer. We need an overpressure or an underpressure, and therefore we even if we have a USP backup for electrical current, we have an additional system with compressed air to keep it fairly safe in the over-under pressure mode. So
these were the first 3 ones. Then this is the 4th one. Actually, you see here, this is the, whoops, that was a little bit too fast. That is actually the point where we started. Actually, this gives, let's say, the view how the Bosch capping machine was looking once before we started the design. And you see it is very condensed. You have a lot of hidden surfaces. And certainly during capping, we have freeze-dried vials, you will have breakages, then you will have powder on the machine table. So we said, I mean, this machine basically, even though it was said by the supplier that this is their isolator version, but we said this is not a good high-potent solution. And therefore we said we have to certainly change the design of the machine significantly. And what we basically did is we, was again too fast, that we separated a part of the machine where basically the cap sorting and charging is there, because this is a part which never gets contaminated. So basically we segregated this machine into two parts, basically the contaminated part and the non-contaminated part. This part is in overpressure. We have a mouse hole here through, so that we can always ensure airflow from the non-contaminated into the contaminated part. And by doing this, we reduce significantly the installation density on the contaminated part, where we then said at the end during the mock-up study, yes, okay, now we are at a level where we can really say this could be cleaned by the operators, of course, where the isolator is still closed. This was very difficult to photograph this, therefore, I mean, you see here, this is then the actual result of it, where you see here, this is really the contaminated part, and this is the non-contaminated part, and as said, we have a pressure segregation here in between these two isolator segments. Another part is, unfortunately, there will be a session here also about robots. Our machine is still working on a conventional way, meaning we have a lot of star wheels and we have a lot of transportation belts. And transportation belts, specifically, if you imagine if the vials are coming from the freeze dryer, they are highly contaminated. First of all, there are residues on the outside of the vial. Then you might have breakage in the freeze dryer, and this breakage you will push out onto the belt system, and it will definitely highly contaminate such a belt system. And with belt system, it's always a point, I mean, they are very fragile, they have a lot of corners, they have a lot of surfaces. How to clean them? And we said, I mean, if an operator would clean them manually, that would be very hard and very difficult to get a reproducible cleaning result. So therefore, we introduced here, let's say, a small tool where we have, let's say, we call it a fork, and this fork, let's say, is spraying on the outside of the belt, on the upper surface as well as on the lower surface. And by validating this kind of cycle with a certain belt speed, we have achieved a very good and a very reliable and a very reproducible cleaning rate of the belt system. Next one, I think this is again yours. Now we are coming a little bit more into the freeze dryer, and as Richard was saying, also the freeze dryer is highly contaminated, and therefore we did also a lot of efforts here in order to improve, let's say, the cleaning of the freeze dryer itself, but also of the technical systems of the freeze dryer.
So this loading system has a vacuum bar that holds with this with these holes, sucks a little vacuum there and holds the vials in this position before they get to the belt and the unloading process. And there we also, because you suck away something, we also had to validate that we can clean it, and you can see the test with riboflavin here. So this is what's below this belt, and I have to add that cleaning the belt would normally mean without this spray, well, that you have to hold the spray ball for 5 minutes in the hand, and I think this is a very important thing. This small investment with this automatic system is really make sure that it's really held for 5 minutes. Good. This is the pusher system for the freeze dryer, and you can see that we have many nozzles that clean this system. And here you can see a picture from the FAT. There we put a plexiglass thing so we could see how it really gets clean. Another thing is, of course, the inside of the freeze dryer has to be cleaned perfectly, and so this is a system of the company GEA with a multi-connection port that makes sure that it's easier to clean. You can see with the arrow, this is a picture of this multi-connection port, and there we also have some additional nozzles that clean this area. Another improvement is we have, I think, the first condenser that is really accessible. Most condensers you cannot climb in, so you cannot prove that there's high potent substance inside. So we designed it with a accessible door. It's 60 centimeters, and you can go in that way. And to prove that really you can really get in, this is Ben Jacobi from NNE, and he really went in, and you can do swaps and really can prove in the cleaning validation that nothing is inside the condenser as well. So another problem is the vacuum system. We said we always want to have 2 filters in row, because if one breaks, then nothing goes to the outside. And then when we told GEA to build this, they said, okay, this the biggest vacuum filter we have. The biggest vacuum filter we have. This one has such a high pressure drop that there is no vacuum pump in the world that can handle that. So what we did is we designed the vacuum pumps in a cleanable way, and then have an additional security HEPA filter as a back-in, back-out filter on the pressure side, and that's something the vacuum system can handle. So this is how it looks in reality. You can see how big this first filter is, and it was also a very interesting technical task. Okay, then we want to do this together. So as Hartmut already said, the pre-engineering study for the main process was very important, and I would do this, I recommend to do this again. And also the FMEA we did a separate FMEA only for the SHE task, because it was our first high potent project in Mannheim. Of course, we did a FMEE analysis for the sterile part, aseptic part as well. And it was very challenging to do that. And we did this with the suppliers. I think this is the very important thing. And what we learned is that every supplier had different solutions. And we wanted to take the profit from this discussions as well.
So if I look at the time, just a few other points where I think it was really beneficial to the success of this project was certainly, I mean, the complete design was building integrated management, so that we had a complete 3D model of the entire process system as well as of the technical systems. We had a shared project office during all of the time, meaning, let's say, internal and external engineering sources from Roche and from the contractors. We were sitting all together in one office, which enabled a much better communication, a much more intensive communication, and therefore also, I think, even in the coffee breaks, there were a lot of good solutions developed during that time, so that helped also quite a bit. And then we had a very sophisticated document management system. You will be shocked if you see the number of documents which were generated during this project, and without these systems, I think we would have been totally drawn in nowhere. Just a few figures which might be interesting. I start with the first one. In total, since the project was SAP controlled, we had approximately 300,000 engineering and qualification hours, approximately 200,000 external, 80,000 internal, quite a bit of construction hours, as you see there. And now comes the big bang. We have these 12,000 qualification documents, including attachment. And since every version was saved, at the end, our server contained 125,000 documents. So a bit of paper. I mean, we didn't print this out, by the way. So I think this was it, yeah. Last but not least, of course, always the coffee point. Yeah. I think it was not only 80,000 cups of coffees. I think we damaged 3 or 4 coffee machines in total. Anyhow, so last but not least, I hope if it is running, there would be a short video, but let's see if it comes, and then Wolfgang will just say a few words on the video that you really see that is.
I just wanted to let it run to get an impression, and we can start the discussion during the time that the video is running.
Right, and we are exactly at 0.0. Thanks very much for listening.
Thank you.
Oh, it's already stopped the video, though. Was it?
They stopped the video. They can let it run.
So you can still let it run when, during the time we have the Q&A session, yeah. But thank you, Wolfgang, thank you, Hartmut, for the presentation. So now we have your time. There is a microphone somewhere, otherwise we might not easily understand what yours. This one is on. Otherwise we would not understand each other.
Hello. Sounds like you can hear me now. For that last example, is that facility designed for multi-product? Is it a multi-product facility?
Yeah, yes, it's also for multi-products, yes. The first products we're producing are normal products, biotech products.
But we have to say everything which is direct product contacting, as Wolfgang has explained, is dedicated or single use. So therefore there is, let's say, no shared surfaces. If you would consider, of course, that a freeze dryer is not a shared surface, but everything which is in direct contact with the product itself, meaning vessels, they are either dedicated stainless steel, product dedicated, or preferably single use. And the same, what Wolfgang has explained on the fluid pass on the filling machine.
It is a lot of investigation, and I can imagine the hours you mentioned there, that this is really also the hours you spend on this, because you have to look on every detail, yeah? You have to consider areas where maybe some product can be and could have a potential risk to your still open containers. And there, when you go through, you can, I think I'm totally agree with you with on the hours what you spent there.
Yeah, I think it was a big challenge to do this facility because it was our first in Mannheim, and of course we had to get into the thinking, and therefore the SHE risk analysis was very helpful because it brought everybody together, and we had to discuss it, and we also discussed it with the suppliers, and this helped a lot.
But it's often not so easy, and that you, also what David said, that when you demonstrate that this could also save costs, yeah, when you implement. I still remember the time back where it was not so easy to make it, where everybody only considered protection of the operators as useless. Yeah. And this is, in general, nothing is useless, everything is important, yeah, because you're a toxologist, yeah. And I always say there is a reason behind that the level is so low, yeah. This is not coming because such a toxologist think, oh yeah, let's give them a nanogram and make then Wolfgang crazy when he has to design it, yeah. So there is a reason behind this, yeah, and often it has a side effect or a lead effect, which not happen now, but maybe could happen a couple of years later. Maybe you have a new.
Yeah, and I'd also like to comment that the toxicologist doesn't just set the OEL, but the PDE. So the beginning of the product carryover calculation is also based on the analysis of the toxicologist. So we're protecting the workers. We're also protecting patients with that number. So it has to be precise, and as many problems occur from over-conservatism as from being too liberal on setting the number. So the pharmaceutical companies spend a lot of time comparing numbers with each other to make sure that we have a good, precise number that's both protective and not wasteful of resources.
And especially when, as also come from the oral solid dosage part, then you often have the discussion, sure, when somebody's inhaling something, does it have the bioavailability that you absorb? But when you become on patient safety, whatever is contaminated in your vial, it's 100% injected into the human body and has an impact. The same with many of those gene and cell therapy products coming to the market, because every contamination risk is even more fatal. You must imagine you gene manipulate from somebody's the cell, and when this is in a small portion coming to the next one, he has those in his body. You cannot say maybe it would have some effect. It will have some effects on it,
Agreed.
There are additional questions? Here.
Yeah, I was just curious in the last project if surrogate testing, something that you guys have looked at or are into or plan for the facility.
No, it's hard to hear that. Can you repeat it?
Surrogate testing, so using like lactose or something like that to run through the line to prove out empirically the control for the containment of the line.
There's an echo when you are on the
Use it without the microphone.
maybe use it and speak
Yeah.
or come closer and use it, because this is such an echo we can't hear it here in front.
Surrogate testing, using lactose and some other surrogate
Very good.
to run through the line and test empirical safety.
Ja, Surrogat, was wir genutzt haben, Riboflavin,
Riboflavin versus lactose.
was ihr benutzt habt.
At the moment, we used riboflavin at first, and then in the PQ we will use with the other methods.
You mean for surrogate testing for operator protection or a good test? In general, there is also an ISP, a good practice guide, assessing the particulate performance of pharmaceutical equipment. In general, it does not cover aseptic processing, but there is mentioned different products. You can use lactose, which normally does not go into the extremely low nanogram levels, but you can use, as mentioned, also sodium naproxen.
Right.
As a one, there is also riboflavin mentioned, I think, and there is also one or two more mentioned in this good practice guide. This is normally the surrogates you use. For this is also important, maybe when you have an appropriate risk assessment, to say, especially in such a large filling lines, where in general exposure could occur, so where you have maybe open process, where you might have some spillage, especially when you maybe unload the freeze dryer, when there is something happen, this is then the highest risk. And this is also location where you normally measure outside. So you measure in the area where the operators are normally working, and also that you positioned air samplers on the close to respiratory to monitor where they walk around and so on.
For me, it was very interesting doing the risk analysis, because I thought the dangerous part in this facility was the freeze dryer, as you say, but we can flood it.
Right.
It's not really a risk. The most dangerous area is where you get off the vials and put them in the tray and get them to the inspection machine. And the same inspecting them in the inspection machine. This is the most dangerous area, not the areas where the persons are protected by isolators.
Also like to point to the importance of continuously testing. So you don't just do an FAT and the validation on site, but then periodically you need a CIH, or board-certified industrial hygienist, to take samples periodically over the lifetime of the product, because gaskets tend to fail. It can build up in places that you're not expecting, so it's very important to have an ongoing system of evaluation.
So I think we are now right on time for the break. So also for a conclusion from my side, so thank you for your participation on the session today. So please join me to recognize all of the speakers. So please take a minute to rate the session before you leave. It's quick and easy. We appreciate your feedback. The introduction is open the conference mobile app, click on events, select today's date and session name, and click on the clipboard icon to rate the session. So the lunch will be provided in the exhibition hall A to D. So, and then take the opportunity to visit also the exhibitors. Woman in Pharma. There you can also buy a bottom from Charlotte. She said she's here before the break, but you will find her, and then she's there, so you can get those buttons from her. So then, following lunch, return promptly at 1:30 p.m. For a continuous of the presentation on facility upgrades, is track 1, and expansion in Salon F. Track 2, aseptic and product development and disposable in Salon G. And here again, track 3, preventing cross-contamination in shared facilities is Salon E. We will have another case study in the afternoon from Japan. We will go into virtual reality, Industry 4.0, and we will also learn about more automatic offline cleaning this afternoon. Okay, thank you.